Thank you very much for having me here, and good morning to everyone. So, switching gears to, I'm going to be discussing malignant diseases occurring in much older populations than fetuses, fortunately. And so today I will be talking to you about my lab's work on studying myeloid malignancies, in particular acute myeloid leukemia, and in particular the clonal evolution of acute myeloid leukemia with induced pluripotent stem cells. So in my lab, we are developing induced pluripotent stem cells from patients with myeloid malignancies to study these diseases. And I'm sure this cell and gene therapy audience is very well, very familiar with the concept of modeling inherited monogenic diseases of the blood or other systems with induced pluripotent stem cells. But what we're doing here is slightly different because we are trying to model a somatic disease, cancer, that is, and mutations that are acquired somatically. And I will explain to you why this has several differences in the procedures and techniques we use for reprogramming, because this is a heterogeneous population of somatically acquired mutations that we are reprogramming. Just as a means of brief introduction, acute myeloid leukemia is a cancer of the blood. It's a very aggressive cancer that will very rapidly kill patients if not treated. And it originates in the hematopoietic stem progenitor cells residing in the bone marrow that normally will differentiate and sustain the blood production of all different mature lineages. But in the case of leukemia, these cells are blocked in their differentiation and instead become very good at proliferating. And so the bone marrow and the blood of these patients gets filled with these immature stem progenitor forms that are called blasts. They have these very big nuclei and little cytoplasm. So if you see this in a slide of a patient, this is really really bad news. And we now know very well that leukemia develops through a process of clonal evolution with the gradual accumulation of mutations by hematopoietic stem progenitor cells, and that this process is also importantly coupled with our appreciation of distinct clinical entities that are precursor stages of leukemias. And these are, unfortunately, this is not working, but, and so we know since several years we have described these entities called myelodysplastic syndrome, or MDS, which has also been referred to as pre-leukemia. It is a condition that that is characterized by a block of differentiation, but not yet the full-blown proliferation that characterizes leukemia. And these patients very frequently are at risk of progressing to AML. And more recently, by sequencing the genomes of the blood of healthy individuals, we realized that a lot of the healthy individuals, especially as they age, contain clones in the blood that are also share some of these mutations that are shared by myeloid malignancies. And this condition has now been called clonal hematopoiesis of indeterminate potential, or CHIP, or aging-related clonal hematopoiesis. It's a condition as frequent as 20% of once you reach 80 years of age. So even though these conditions are not necessarily succeeding each other, so you can have a leukemia without all these preceding stages, or at least not in a way that are clinically recognizable. However, we know that they are related because they share a common set of mutations. So And so we know that, for example, this is very strange. For example, in clonal hematopoiesis, you have one mutation most frequently and very rarely more than that. We know that in MDS, you have 2 or more mutations most commonly, and in leukemia, more frequently you have 3 to 5 mutations. And by sequencing those, the genomes of patients at these different stages, we have been able to characterize this sort of mutational landscape to mutations that tend to occur early in the disease and mutations that tend to occur late in the disease, and also mutations that occur at intermediate levels, or they can occur both early and at later stages. And so all this is now give us the opportunity to ask many interesting questions about the clonal evolution of leukemia and cancer in general. However, once you have a leukemia, most frequently you have all these different clones in the bone marrow of the patients, and you can't separate them and study them to understand what happens to the cells in each step of this process. So in earlier work in my lab, we hypothesized that because reprogramming allows us to capture cells at a clonal level, that we could potentially take bone marrow of patients with myeloid malignancies, like leukemia and MDS, and by reprogramming the cells of the bone marrow and blood and using their genetic composition of mutations to understand the provenance of these clones, and I will explain to you in a second what I mean by that, we could capture different clones at different stages of the disease and model this process of clonal evolution. And so this is what we did, and it actually turned out, before I get there, even better than we thought, because it turned out that reprogramming actually favors, even though in advanced stages of leukemia, the bone marrow is filled with the more advanced malignant cells, reprogramming usually favors more normal cells. So even if these more early stage cells are rare in the starting cell population, we can get them out much more efficiently. And so what we did is we started reprogramming patients, and for example, I'm going to show you here some examples. We reprogram cells from a low-risk MDS patient, and again, we used the mutations to confirm the provenance of these cells from the malignant clone. This is very important because reprogramming erases all the epigenetic marks, so it is only the genetics that we have to go by. This is a second patient with high-risk MDS, and in this case, we could make iPSCs from malignant clone, that is the blue, but also from normal cells, and this is actually also something that very often we have the opportunity to do with reprogramming these patients. This is great because we have genetically masked controls. And this is a third patient with high-risk MDS, and in this case, this patient had a familial form of predisposition to MDS-AML that is characterized by germline GATA2 mutations. This is actually quite interestingly something that the family or his doctor didn't know, and we basically discovered it by reprogramming because we got clones that had only this GATA2 mutation and none of the other set of mutations. So we went back to his germline by looking at stromal cells and finding this mutation. So this parenthetically, but importantly, these were represent now cells with a predisposition to familial predisposition to MDS and AML. And finally, we obtained iPSCs from a patient that had transformed to AML now. And also interestingly, as you see here, we got clones that had both the deletion 7q, which was the clonal genetic lesion in this patient, as well as clones with both the deletion 7q and a KRAS G12D mutation. There was a subclonal mutation. So again, showing proof of principle, you can get separate different clones by capturing them into iPSC lines. So having now all these different lines, we could put them in this order and have all the stages of the clone evolution of leukemia. So then we asked, okay, can we then capture phenotypically a progression, a disease progression with these cells? And the answer is yes, and how do we know it? I'll show you in the next 2 slides. So to do that, first we need to differentiate these iPSC lines into blood. And we use for this a protocol that is largely derived by Gordon Keller's lab work together with Andrew Elefanti's spinny bee method. So we basically make spinny bees, and by using combinations of cytokines, we coax these cells to make mesoderm, hemogenic endothelium, and then hematopoietic progenitors, mostly myeloid progenitors. And this is just the middle panel, how we follow this, just very simply with 2 markers. CD34 marks a hemogenic endothelium transition into hematopoietic cells, and this is basically this counterclockwise movement of the cells, and I'll get back to that. And normal cells will stop differentiating and become fully mature, mostly monocytes in this particular condition, so we don't want them to become anything else, and stop dividing. Other assays that we can use now to characterize the cells, and that we used, and as I will show you, to characterize this panel of different stages of cells, are basically assays that we use for ex vivo cultured hematopoietic cells from healthy individuals or patients. And these include clonogenic assay on top of flow cytometry for immunophenotyping, clonogenic assays in methicellulose that give us the differentiation output, lineage potential of the cells, morphology. We can direct them to downstream maturation to specific lineages, and we can also try to transplant them into NSG mice. Now, you're probably aware that human pluripotent stem cell derived hematopoietic cells are not capable of engrafting NSG mice. However, we found in this study, and I'm not going to show you the data because it is published, that hematopoietic cells derived from iPSCs that come from leukemia patients can actually engraft robustly a leukemia. And similar findings were also described at the same time by Ravi Marchetti's lab at Stanford. So it seems to be at least the case for several AML iPSC lines. Okay, so using now all these assays, we were able to determine that indeed these cells capture a phenotypic progression, whereby we have a gradual decrease of differentiation and initially proliferation, and then a proliferation increase again. And this is here in more detail, but this is a summary of the data. I'm not showing the data because it's published, but basically we see a lot of cellular phenotypes that we know already of from studies in patient cells. For example, we know that MDS is characterized by cells that are blocked in the differentiation but grow poorly, and then once you get leukemia, this is restored and you start proliferating. And importantly, we also know that we also found that these leukemia cells engraft in mice, which is something that is very unique to the leukemia stage. We also found that you can take iPSCs from a high-risk MDS stage and correct their lesion that is for example, in this case, the deletion 7q, and revert them back to a pre-leukemic stage. And we showed that you can do many different things like that by playing with gene editing. You can introduce mutations in a pre-leukemic stage and move the cells to a low-risk MDS, for example, by inactivating the other GATA2 allele in this particular case, or by deleting 7q and moving it further down to a more aggressive high-risk MDS. And we can also take normal cells, move them into a pre-leukemic state and further down to a high-risk MDS. So, and finally, we also found in this study that these now hematopoietic cells differentiated from these different iPS lines captured in these different stages have signatures that correlate with these phenotypic findings of progression, but that also they have, by gene set enrichment analysis that I'm showing here, gene signatures that are found in primary leukemias. So this earlier study taught us a few very important things. One is that indeed we can use reprogramming to capture different stages of leukemia from patient cells directly. That these lines can capture a phenotypic progression, and that gave us a roadmap, a framework of cellular phenotypes that we can now use as assays. This is very important in particular because there's, as I'm sure many of you are aware, iPS-derived cells are relatively developmentally mature, and they can capture different developmental stages. And this creates an uncertainty as to how much of an adult phenotype they can capture. So for us, it was very nice to see that we can really capture some phenotypes that are really disease relevant, reproducible, and that we can use as readouts. Also importantly, we found that once you differentiate these cells back to blood cells, they will re-establish gene expression signatures that are found in leukemia as we have found them in primary cells. And then you can use CRISPR to move these cells back and forth different stages. So all these were very important for us to move forward. But one limitation of this type of study and this set of lines was that all these different iPS lines were created in different genetic backgrounds. They came from different patients. And so even though we could find some gene expression signatures that were shared with primary cells, that was not sufficient to go into like a discovery mode and interrogate these transcriptomes or epigenomes of these cells to derive more general hypothesis as to what happens in the different stages of transformation. And so this prompted us, gave us the idea that we need to make this model into a common genetic background to eliminate all this variation. So we thought that maybe we can do this. Well, one way we could have done this would be to take patients in longitudinal samples and try to reprogram from one patient all these different stages. However, this is uncertain in its feasibility because for different reasons you may not be able to get a specific stage. But also these different stages in a patient are, there's many cell divisions that are required to go from one stage to another over many years. So these cells are really, have many other different passenger mutations potentially and would not be strictly isogenic. So we thought that maybe we can do this by just taking normal cells and building our own leukemia from the bottom up, so a synthetic leukemia. And I think this is something that would resonate very well with this audience, and I am a genetic engineer at heart, so I think that that really like the ultimate best way to understand cancer is to just create it in the lab. But that is not something very trivial, especially not with human cells, who are fortunately much more resistant to be transformed in the lab than mouse cells. So several studies before have attempted something similar in other cell types. Work mostly by Rob Weinberg's lab has actually succeeded in transforming primary human melanocytes or primary human astrocytes, but this work used very strong viral oncogenes that are not really found mutated in spontaneous human cancers. And more recent work succeeded in transforming human prostate and lung epithelial cells with now with oncogenes that are really relevant to these cancers. But again, these used lentiviral overexpression to express these oncogenes. So we thought that maybe we now have an opportunity to create better de novo oncogenesis models with human cells. We have iPSCs that are very amenable to very precise and controlled introduction of specific genetic changes. We have, fortunately, CRISPR that has opened all these new possibilities to do this fairly fast and efficiently. And we have sequenced so many cancer genomes that we know a lot about the mutations that are at play and that are needed. And as I told you a little bit in my introduction, we also know which mutations occur at what different stages or tend to occur at different stages. So we know a fair amount, and especially in leukemias, we have the additional advantage that leukemias are very simple cancers. Here is, again, a graph that you may have seen many times if you care about cancer genetics. And you can see that basically from left to right are different cancers with the numbers of mutations they have. And as you can see, leukemias are all the way to the left. So they're very, very simple cancers. And as I mentioned, you can have a leukemia with as few as 2 or 3 mutations in a normal karyotype, which is in stark contrast with solid tumors that have typically tens or hundreds of mutations, a lot of them presumably non-drivers, but still it's very hard to figure out what is really needed and what is not. We have a better idea to do this in leukemia. And so a brave postdoc in my lab, Tiansu Wang, who joined the lab 3 years ago, and who was already a genome editing expert, said to do that. And so he decided to take normal iPSCs and start introducing mutations using CRISPR one by one with the hopes of recreating a clonal evolution model. And so, so then we asked, as I showed you before, we know already a lot about which mutations tend to come early, later, or very late in the disease. And these tend to be, early mutations tend to involve chromatin regulator genes, and late mutations tend to occur to involve genes involving cell signaling. But we also know even more about how these different mutations prefer to occur together with each other. And this is work by our collaborator, Ellie Papa-Emmanuel, and she did most of this work at the Sanger and Peter Campbell's lab at the UK, and now is in New York at Memorial Sloan Kettering. And what basically they did in this sort of landmark New England Journal of Medicine paper published recently is that they sequenced many genomes, and that gave the statistical significance to say which mutations prefer to occur with each other and which mutations tend not to occur with each other. And from this, they could derive potential mutational paths in leukemia. And so by discussing with them, we decided to take these 2 paths that I'm showing here. One is short name NPM1, the other is chromatin spliceosome, as the 2 most common mutational paths. And NPM1 is the potentially, so this group has DNMT3A, NPM1, and FLT3 mutations, and it is the most common 3-gene co-mutation combination in leukemia. And the other one involves a combination of genes regulating chromatin and splicing. So more specifically, the genes that we picked for the first root, the chromosome spliceosome, and this is a root that almost always involves a pre-existing MDS stage before the leukemia, was A601 C-terminus truncation as the first mutation. Again, this highlights why doing this with CRISPR is so much better, because we don't really know if this mutation is a loss of function or gain of function mutation. There's studies that are contradictory. So if you were to model it in some other way, it wouldn't be obvious how to do it. So we basically modeled one of the exact C-terminus truncations that are found in patient cells. As a second mutation, we picked the SRSF2P95L mutation. This is a splicing factor gene. And as last mutation, the NRAS-G12D mutation, and this is, again, a signaling mutation. This is a very common late mutation in leukemia. For the second path, we picked DNMT3A as our first mutation. This, again, is a DNA methyltransferase. It's a very common first mutation. A second NPM1, and then FLT3-ITD, again, a signaling mutation. And while we're doing this, a foundation, a family with RUNX1 familial mutations, germline mutations, started a foundation and gave us a grant to also build their progression to leukemia, the personal leukemia, by starting with the RUNX1 mutant cells and adding mutations. In this case, we decided A601 and FLT3-ITD. The thing with familial forms of leukemia is that, unfortunately, we don't have that much data on coexisting co-occurring mutations, because these are such rare cases. So we don't have the statistical power to say for sure what are the mutations that prefer to happen more frequently in these patients when they progress to leukemia. So the data I'll show you in the next 3 minutes are from this first root. And it's my mistake, I should have changed this slide, but we actually also added a fourth mutation in FLT3 to give it a higher chance of, becoming leukemia. So just a small technical slide of how we do that. And this is showing the step of going from the double mutant to the triple mutant, and then the triple mutant to the quadruple mutant. So we basically nucleophect our cells with the Cas9 RNA vector and donor vectors. And importantly, in the case where we want to introduce a mutation in a heterozygous state, which is not very trivial, we have found that it is much more efficient to actually deliver 2 donors, 1 wild-type donor and 1 mutant donor, and then we don't care about clones that have indels, but we're just focusing on the biallelically edited clones and select those that are heterozygous for the mutation. So that has worked much better for us, and has helped us make a lot of heterozygous mutations much easier. So then we screen single clones and by RFLP, and then further characterizing them. And so Tiansu did a very careful, tried to be very careful in each stage, find clones that were very well characterized, not outliers in any phenotypic assays, and then move to the next step. So that took Tiansu over 2 years to do. And so then we're very, of course, interesting to see, was this worth it? Did we actually make something that looked like a leukemia? And it seems that we did unfortunately. And so I'm showing you here the progression of the cells during differentiation, and this is a snapshot of day 14. As you can see here, and this is also shown in the graph to the right, there is a gradual differentiation block, and as the cells go from one stage to the other, they actually get more stuck in the 34 positive stage, and they can't move on to more mature stages. And this is shown here also in terms of morphology. The cells to the left have more cells with morphology of monocytes, this large pale cytoplasm, and as they move on, you see more immature forms with large nuclei, more like blast-like cells. The same is here showing in colony information assays, differentiation, gradual differentiation block. Remember, in the patient-derived cells, we have seen that you have a slower growth up to the MDS stage, and then that gets restored, and you proliferate like crazy, like a leukemia. And this is exactly, again, what we found here. And to the left, and then by same thing by cell cycle analysis. And in the lower left panel, we found very interestingly that not only the cells had the increased proliferation, but they also had the ability to expand in culture for longer. So they would not go into terminal differentiation and arrest the growth, but they would keep growing. And I guess even more convincingly, when we took the triple mutant cells and the quadruple mutant cells and put them in mice, they indeed transplanted a myeloid leukemia, and this is something that, again, as I said, that no cell that is not a leukemia iPS-derived hematopoietic cell can do. So this was made us very happy that we can indeed make something in the lab that looks like a leukemia. So what we are, and furthermore, when we looked at the transcriptomes of these cells compared to normal cells, we could find, again, gene expression signatures of primary leukemias. So what we are now doing is analyzing transcriptomes and chromatin of these cells with chromatin accessibility assays like a TAC-seq. And these are some preliminary findings. So we see things that mimic or mirror the phenotypic progression, so changes up and down that tend to accumulate as you accumulate mutations. So we're now digging into this data, and I'm going to stop here, and trying to understand, try to find transcriptional programs or other epigenetic events that correlate with progression that may be important in causing this, and that we can potentially use to intervene to either halt or prevent or predict this progression. So this is the summary, and I already mentioned this. And so the earlier published work that I presented was mostly the work of Diana Cotini, a senior postdoc in my lab who's now an instructor in the lab, and CJ Chung, who is a former member of the lab who's now at Regeneron. And the synthetic leukemia work was done by Tiansu Wang. I mentioned our collaborators, and we have several funding sources, very expensive work, and we are currently recruiting. So if you are interested in working in the interface of stem cells and cancer and genome editing, of course, and genomics, so please send me an email. So thank you very much, and I'm happy to take questions.
Any questions? We are out of one question, quick question.
Yeah, I was wondering how stable the mutations are in the iPSCs, how frequently you monitor them, and also can you do that at a single cell level?
Well, okay, so the mutations are stable as mutations are stable in cultured cells. So I mean, they're not less stable than they would be in any other cultured cell.
The iPSCs have unstable genes.
That is not true. That is absolutely not true, and this is something that is repeated again and again, that iPSCs have an unstable genome, and there is absolutely no evidence that iPSCs have an unstable genome. There's actually a lot of evidence that iPSCs do not have an unstable genome. There were early, just with this opportunity, I'll take just 10 seconds. There were early studies suggesting that because you sequence the starting cells, you find, I don't know, X number of mutations, then you sequence the iPSCs, you find more mutations, that this somehow miraculously arose during reprogramming. But then many subsequent studies really show that if you go deep and look at these mutations, try to find these mutations in starting cells, you will most frequently find them there. And this is because reprogramming, as I said, is a clonal process, so you pick one cell. So, the cells are mosaic for many, many different mutations, so you can pick a clone and expand, suddenly a set of mutations that seems to be new, but it is, and if you go deep, you will find it. So iPSCs do not have unstable genomes, and please do not perpetuate that. There's many studies now that have shown that iPSCs accumulate mutations in the rate of division that is predicted by just cell division, like any normal cell would do. And of course, iPSCs, like every other cell that you culture in the lab that has the potential to be cultured for many, many, many divisions, you do, of course, need to monitor for genetic changes that will either caused by genetic drifts or by a selective advantage towards to expand a clone or another. And the second part of your question was if I have time.
Yeah.
No.
How about we?
No, Dan. No, I don't want to take your time.
We are
Well,
about
I'll
10
answer
minutes
them.
late. Then this.